Hi, we're Group F, and this is our presentation on how evolutionary canyons help provide insight into cyanobacteria adaptation. My name is Neil Ragland.
I'm Brian Lopez.
I'm Lisbeth Caballero.
I'm Colton Lowney.
I'm Isaac.
I'm Simran Sharon.
The two articles that we decided to focus on were our first article being about evolutionary canyons and how the microclimates and the microclimate contrast of each of the canyons helps with the bacterial adaptation. And our second article was about lateral gene transfer and its effect on bacterial innovation and adaptation. And our third article combines these two and talks about microclimate stresses on the adaptation of a specific species of cyanobacteria found within the evolutionary canyons.
So the article 1 is basically about the adaptation and incipient sympatric speciation of Bacillus simplex under microclimatic contrast at the evolution canyons 1 and 2 in Israel. So basically it's 945 soil isolates of Bacillus complex, and they're on two different slopes of the Carmel and Galilee. They're located in Israel, and then they're separated by about 40 kilometers, but they both provide similar conditions. So basically each canyon had a sun-exposed African-like slope and a shady European-like slope. And so what the main findings from this study was that the strains representing genetic groups were identical in the 16S sequencing, meaning that there was high genetic similarity and monophyletic origin. So basically they all came from like the same place. Ecological contrasts were more heavily correlated with parallel and phylogenetic structures than was geographical distance. So basically, whatever was going on in the environment had more to do with what genes came about rather than the geographical distance between the samples that were there. The slope-specific populations differed substantially in regards to diversity, meaning there was just a lot of different types of genes that were available and present. So basically they showed levels of DNA repair, and they measured that with UV sensitivity, and then mutation rate was the resistance to measured mutation rate by their resistance to rifampicin, and these heavily correlated with ecological stress and phylogeny. And then like the main takeaway was that ecological conditions / the stresses that these populations go through are the driving force for the diversity within their population, not so much geographical distance. And we kind of already know that. This is just kind of like giving us more examples of, you know, your environment plays a big influence on your genes as well as your phenotype as well as your genes. And then it's also just natural selection kind of promotes those that are more suited to combat the stress that's in the environment.
So a summary of Article 2, lateral gene transfer in the nature of bacterial innovation. So we know that horizontal gene transfer or lateral gene transfer is the transfer of DNA from one bacteria to another. There are three mechanisms in which lateral gene transfer occurs. That is conjugation, transduction, and transformation. For conjugation, there needs to be physical contact in which genetic material is transferred from donor to recipient. In transduction, the bacteriophage can insert DNA into a recipient. In transformation, it is the uptake of genetic material from dead bacteria around in the environment. So, through lateral gene transfer, organisms can develop traits. These traits include antibiotic resistance, which allows microorganisms to expand their ecological niche. So they expanded to new areas and obtain new genetic materials, which is beneficial for them. Trait number 2 is virulence, which is that allows for adoption of pathogenic lifestyle. This usually involves a fundamental change in the microorganism's ecology. And lastly, metabolic properties, which allows microorganisms to obtain new mechanisms. When these new mechanisms are obtained, microorganisms can explore new environments. This emphasizes the importance of natural selection.
With our third article, it focused on a specific species of cyanobacterium that resides in the evolutionary canyons called Gnostic Linkia, if it's pronounced correctly. And the purpose of this article was to talk about a study that they performed, which was testing two hypotheses. The first hypothesis was the niche width variation hypothesis, which was also coupled with the environmental theory of genetic diversity. And what this was really trying to get at was they're trying to find if the theory was correct about populations in highly changing environments, such as the microclimates found in the evolutionary canyons, affected the polymorphism of a bacteria species. And then they also wanted to see if the second theory, which was called the neutral theory, also applied to these bacteria in these environments, seeing as in this theory, it suggests that a large population size with a high mutation rate should have an increase of nucleotide variation, which should increase the high polymorphism of that they're looking for. What they target, the genes that they targeted for finding if these hypotheses were supported were called the Chi genes, specifically the Chi ABC cluster. And these genes affected the bacterium with it affected its rhythm, a circadian rhythm, and how it functioned during its life during in the evolutionary canyons. To test this, they had to separate the cluster from each other since it was a single copy, meaning they had to use PCR, and then using the PCR primers at specific locations in the DNA, they amplified these sections, took these amplifications, purified it, and then these purified selections underwent gene sequencing.
The results from Article 3 show that the Chi ABC family is a monophyletic group resulting from multiple duplication events caused by the extreme microclimates that are present in the evolutionary canyons. However, they also noticed that the Chi B and C genes from this family are the most amplified in their gene family, which they believe is responsible for the high polymorphism exhibited by the cyanobacteria. Their research also revealed that the Chi family gene has 5 subfamilies within them, but two of particular interest are the Chi 4 and Chi 5 families because these are found in the cyanobacteria in the evolutionary canyon. They exhibit high duplication rates from the constant UV radiation, resulting in mutations and causing duplication to occur in order to prevent and fix these errors from happening again. They also came to a conclusion that free selective pressures play a strong role in their evolution, which are positive, diversifying, balancing, and purifying selection.
So in the next 2 slides, we'll be talking about how Article 1 and 2 correlate with Article 3 by showing how the driving forces and contributing factors of evolution and adaptation under natural ecological conditions are occurring in bacterial population.
So the primary similarity between Articles 1 and 3 can be seen in how the experiments were conducted, primarily the conditions which under they were conducted. And that's what we've talked about so far as the evolution canyons in Israel, the African like and the European like. And what's also so cool about them is they provide evolution under natural conditions at a micro scale for bacteria compared to laboratory conditions. So they both focus on cyanobacteria, and the goals are also quite similar in that the goal is to gain a better understanding of the driving forces of evolution and also what factors do and don't have major effects on evolution under the contrasting ecological conditions, being the different slopes, sun exposed and shady. And what the results show is that the level of genomic diversity was directly correlated to ecological conditions. Some more drastic ecological conditions, more harsh, the sun exposed, showed higher diversity where the bacteria was residing. And factors such as neutral mutations are neutral being inheritable, so vertical gene transfer, while it doesn't play a role, it's not a major one. Same with geographic distance. When compared to ecological stresses, selective pressures, it doesn't even compare. And also the approach was quite similar in the ways that the focus wasn't morphological differences, physical differences, it was genomic, which provides a much better insight into evolution. And that's it.
So to correlate between Article 2 and Article 3, lateral gene transfer and evolution helped the cyanobacteria to evolve in harsh environments. So since there were two canyons, both canyons had two different types of conditions. One of them was a cold and temperatures one, more forest-like, which was the European side, and the other one was African slopes, which was more exposed to UV radiation by the sun. So they had to evolve in these two types of conditions. So with the help of lateral gene transfer, they use adaptation. So when these cyanobacteria were exposed to the environmental stressors, they had to go through a genomic change in their genomic structure, which allowed them to develop new traits. These traits allowed them to survive the cold conditions in the European side and the harsh conditions that were exposure to the UV radiation. So this allowed them to survive and adapt and also reproduce and increase their bacterial population. So that's it for our presentation. Thank you. And if you have any questions, please let us know.